Muchas gracias. Bueno, ¿se oye bien? Bueno, creo que vamos a empezar ya con el seminario. Voy a presentarles a ustedes al profesor Fuchse, que es la persona que nos va a dar el seminario. Well, now can you listen me? Well, it is for me a great honor to introduce again to this audience, and particularly to those of you who had missed the opportunity to meet here Professor Kjell Fuchse from the Department of Neuroscience at the Karolinska Institute in Stockholm, Sweden, with whom I have had the privilege to collaborate in several topics of neurosciences for nearly 50 years. Professor Fuchse, who is a kind of legend in neuroscience, obtained his MD at the Karolinska Institute in 1971 and remained there as one of the more distinguished and influential members. There, he has been part of the Nobel Assembly, the committee which selects the Nobel Prize winners in medicine or physiology, and is now Professor Emeritus within this scientific institution. On the other hand, Professor Fuchse's scientific career has been characterized by a number of exceptional discoveries, which started with the demonstration for the first time of the localization within the brain of the monoaminergic neurons and tracing some of their pathways, giving rise to the born of the chemical neuroanatomy. Later, his efforts paved the way for flourishing the neuropsychopharmacology. And as a matter of fact, within this discipline, the work of Professor Fuchse with bromocriptine has been one of the more important accomplishments. He discovered the mechanism of action of this important drug. Where he discovered, his discoveries were tremendously important for understanding Parkinson's disease, schizophrenia, affective disorders, and addiction, among many other brain disorders. More recently, his work has emphasized the importance of volume transmission as a new mode of synaptic transmission and its modulation through the allosteric interaction of neurotransmitter receptors within homo and heteroreceptor heterocomplexes. I must say that in addition, that thanks to his tremendous work, that is not surprising that along his scientific career, Professor Fuchse has been appointed as Doctor Honoris Causa from several prestigious universities, has been admitted as corresponding member from several scientific academies, including the Mexican Academy of Science, and had received numerous prizes and honors. Finally, for all of you who love statistics, it will be rather interesting to know that Professor Fuchse has published his work according to the Semantic Scholars in 1,664 articles, which can be cited 91,664 times, belonging to the select group of highly cited members of the IC Web of Science, with an H index of 139. I leave you with Professor Fuchse. I am ready, but perhaps you would like to hear.
Okay, good.
You told me that you perhaps would rather prefer to be downstairs. Well, now I go upstairs.
Yeah, okay. Thanks a lot. Warm thanks. Perfect.
Yes.
How do you open this up?
Don't need this one. You have your own here.
Oh, this works. Oh, that's great. Well, first of all, genuine thanks to Miguel for his presentation of what I have done. But you have to remember that I am a very lucky man. I have had wonderful collaborations with lots of outstanding scientists in the States, in Europe, in Mexico, and I'm very, very happy for that we have had such a long collaboration with Miguel. It has been highly rewarding for us. And and of course, at this moment, I think it's also appropriate to sorry for but I can now how do I show here? Yes, there it is. Oh, sorry. Well, yes, I would like to recognize Professor Pires de la Mora and also the outstanding contribution by Borotto Escuela, Daciel Borotto Escuela. And I will be trying to not to have too many things, but we'll start with something of the past, and that is the yes, there it is. Well, you heard about the monoamine pathways and that we were able to really observe the noradrenaline, dopamine, and serotonin, and all their projections. And the bottom line is that it emphasizes the lower brainstem, because that's where all the cell bodies and the dendritic components are present. And then you have you have projections into the spinal cord all the way down, and you have projections upwards all the way up into the cerebral cortex. So it underlines the impact of the lower brainstem in modulating spinal cord function and also the telendiencephalon, and reaching monosynaptically, which no one believed in, the entire cerebral cortex in various manners. And I think this thing we reported in '65, we could only publish in 2007 here, because it was impossible to publish. You just could publish black and white in those days. So this gives you the background of the work I did in my thesis, and now I think I just give you an example of the. I seem to have problem with pushing this red stuff. Yeah, there it is. So this was just as an example how it looks like here in the dorsal raphe. You see the multiple types of groups of serotonin and nerve cells, which have a major role in innervating almost the entire telendiencephalon. And we were not so strong with the terminals. We could see the terminals where the spinal cord, but I guess we only saw about 10% of the terminal networks in the brain. And I think the real mapping of the serotonin nerve terminal networks in the brain was made by Steinbusch in 1981. I just wanted to recognize that. And of course, these, when I saw these the first time in the lab, it changed my life in many ways, actually. So let's go to the next one. Thanks a lot. This is just to illustrate before moving into the Parkinson's disease story, that here you, in the thesis, we also could see the dopamine nerve cells in the nigra, and very beautiful, and also to illustrate, and with the partial removal of dorsal striatum, we could see the axons moving into the dorsal striatum, but they could not reach into the cortical. They stopped in the dorsal striatum, and the terminals, in the dopamine terminals, we had a problem because they were so easy for diffusion. So you just saw a diffuse dopamine fluorescence developing, but we could. Okay, I think I wanted, no, so we skipped that. I will show some more beautiful slides. We did see the dopamine terminals when we were very lucky with the Falk Lab technology. And so let's go on now. And then I think what I would like to move into now are what we have been working on the last 20 years at least, and that is understanding the integrative activity and in the CNS, and we were aware of in the late 70s, and that we became interested in trying to see if all the neuropeptides that were demonstrated all over the brain, are all these neuropeptide pathways, and if they could interact in some way with all my monoamine pathways, because they were both there. And then we started this story in that way, and what we did was to study how neuropeptides, as CCK, substance P, neurotensin, could modulate the binding characteristics of the various the dopamine, noradrenaline, and 5-HT pathways, and over the neuropeptides subtype receptors. And I think what we did see was that there was indeed a change, especially of the affinity of the dopamine and serotonin receptors. This represented sort of a possibility for receptor-receptor interactions playing a role in the CNS. We even had a symposium on it, and but nobody liked the idea very well, because I think they felt that the effects we saw on the affinity were not strong enough, large enough to be of interest. But we claimed anyhow that there is an amplification on the way from the membrane into the intercellular part. And we spoke about the subheading, a new intramembrane integrative mechanism. And then I think this was in 1981-82, with a review in 1983. We realized that there was no great belief in it. And we realized we had to do a better hypothesis than just physical interaction in the membrane, in the plasma membrane. So then we came up with this type of hypothesis, that if you have receptor A, receptor B, and they can then just not sit around independently as monomers, but they can actually come together in this receptor A, can form a homomer here, homodimers, like that. And then the B, this is A and this is B, you can form another homodimers. And then we proposed that this could be the mechanism for the receptor interactions we observed, that they also can be an affinity between the A receptor and the B receptor. So that was the beginning, and this was in 1993. And since that time, we have been, and also other groups in the world as well, trying to understand if one could find a way to understand these receptor-receptor interactions in the membrane. And then, we can go back. No. Yes. Finally, in 2010, we had a meeting on the changing world of GPCRs in Stockholm. And we then came up with, and I show this because it was the first time that most of the participants, or the vast majority of them, believed in the story, believed that this may have relevance and be true. And then, just wanted to say that the people in the field were there, so that was nice. And you can see then that you have then the homomers, homodimers, and the higher order complexes. And here you have the protomers coming up here, and you have also here high order complexes formed, and they can be the triangular or linear type. And there was also general agreement that one has to call these complexes, we have to give the name, we have to add a heteroreceptor complexes or homoreceptor complexes, because it was discussed that proteins could also interact with these dimers or trimers or whatever, and therefore, to give a more correct image of what was going on, it's better to speak about complexes. So what I will speak about now then in the talk is the hetero. Yes, okay.
There is an earthquake.
Oh, an earthquake. Okay. Yeah, no problem.
Don't worry.
So we can be inside or have to go outside?
We'll have to go out.
Okay, okay.
I'll go with you.
Yeah, okay, good.
Don't worry, it's just
Yeah,
time. We have time.
yeah.
Just a warning.
What, just a warning, yes.
Warning.
Well, that's,
Earthquake might come.
yeah, yeah,
Yes.
yes,
Better be safe, don't worry.
yeah.
This way.
This way, yeah, yeah, yeah. But you have had earthquakes here before, right? Or no?
No, never in a talk.
No.
You can say in Sweden, Mexicans were so impacted by the complexes that there was an earthquake.
Yes, yes. Hello, nice to see you. Yes, good to see you. Good to see you.
We should have met 20 years ago. Francesco organized a dinner, but I didn't go.
No,
but
no.
finally we met.
Yes,
So nice to meet you.
Nice to meet you as well.
Yes,
yes.
I'm so glad to see you so well.
Well, I'm still alive, so.
Yeah. So you went to the ceremony meeting in Cancun.
Yes,
it was
I was in Cancun.
And travel with the Trevor Sharp.
Yes.
Sharp. Yeah, he was coming to visit me and he didn't reply.
Okay.
And I didn't realize that the Cancun meeting is already there.
Yes.
I was going to go to the Cancun meeting. I saw him in Oxford some months ago.
Yes.
So nice to meet you. How many days you will stay here?
Well, I will leave on the Monday.
Monday?
Yes.
Monday.
I
Oh, I hope you can visit us.
Yes, that would have been wonderful, but I have to go on Monday. There is no alternative, But it's great to see you. Instead, I would appreciate if you could send me one or two of your recent papers, or just what you think.
And our paper was quite good. The paper we did in it's quite highly cited. I was so happy because you are so famous. The amygdala with the the interneuron story. That was very well. And you helped me in the last stage to remember you sent some message to the editor.
Yeah. Well, I think, I don't know if that was actually a real earthquake, but in any case, we made an exercise, a simulacrum.
Good.
So now I think that we have to continue with the talk of Professor Fuchs, and then I leave you, Professor Fuchs, and ask you to enjoy this stuff.
Yeah.
Yes. We continue. Like in theater, this was the circle. We continue.
Yeah. Well, I was something very special this, but certainly I will try to cut the lecture a little bit because we are getting, we lost some time here, and I don't want to to ask for too much time. Oh, but this was for us then an important event, and now I will. So then I just wanted to indicate to you about this complex, that if you go into the most simplest ones, a heterodimer, what do we really know about them? They have not been possible to crystallize them, and we did oh, now this come, but then I skipped this one, the other one then, so we go straight for this. No. Yes, this is the one. The model, to get the model of the, in this case of the heterodimer was the A2A D2 receptor heterodimer. And what we got out of it is illustrated here. And you can see by means of, we had an expert on docking experiments. Jens Carlson was with us. So he was the leading guy on these aspects. And what came up was this type of model. And you can see that in the interface here, it looks very similar to especially this chemokine receptor over here, when you have the transmembrane 6 and 6, 5 and 5, 4 and 4 coming, and you have exactly the same in our model coming up like that. So also we had a large buried surface area in our model, and also in these other crystal models of homodimers. These are homodimers. This is the first heterodimers coming up. And it was nice to be able to turn, this is the D2 and this is the A2A receptor. You can turn them around in 90 degrees and show the surface of the interface here and here. And also, I don't want to go into too many details, but this is just to indicate what you can get out of this type of modeling. And that is that in the docking experiments, there was an indication that in the D2 receptor, in the transmembrane 5, these residues were, residues over here were of interest, and then we did notation of them, of these two, and then we looked and with molecular dynamic simulation snapshots, what happened, how did it look like, and it seemed to be coming up a white part over here between the A2A and the D2 receptor as a result of these mutations, and also the side chains came apart, especially look here again. So this is just to give you an example that in order, it is a lot of effort and a lot of work. So if you want to do something about this, you should select one and do a good job on that one, or discover a new one. Because it takes a lot of effort to do it. So that is the first model we did, and it was the A2A D2 receptor, which is important, especially for Parkinson's disease, in my thoughts. And then you wonder, of course. You wonder if the what gives the affinity, what makes the D280 receptor come together then? What can happen? And it is a difficult thing to establish that really bonds and clasps develop. How can that be true? And we have an hypothesis that still may be correct, perhaps, who knows in the future. But what we proposed was we studied 48 pairs of receptors with our colleague Taracano. He was an excellent bioinformatic expert and also mathematically trained. And what we found was that in the one that did, of all these pairs of receptors of GPCR, there were some that were actually having something interesting, because we believed in that the homomer could play a role. And what we found is that we found sets here of triplet amino acid homologies that could participate in a good way, in an important way in the receptor interface. And that they in some way give a code for bonding and for clasping or clasp. And we also called it the hotspots, perhaps. We will see if this hypothesis will be true or not. We have not been able to do all the necessary mutations to really establish their impact. But you can see here we have 4 different types of we call them the triplet puzzle, actually we called it. And you can see here that you have 4 different types of homomeric triplets coming up in blue, in the D2 receptor, in red, in the A2A receptor. And here they are in the transmembrane domains. Here they may play an interesting role coming up here, and we did a similar analysis with neurotensin 1 and D2 receptors. You can see these actual triplet homologies coming up also here. In NTS, they are red, and over here they are blue. So it may be, when we have appropriate work has been done, that they could be important hotspots in these pairs of receptors coming together. And here, 4 of them here and 3 of them over here. So we feel that this could be one possible way to understand what's going on and why they stick together strongly. Okay, and now we have the next one, and what I wanted to indicate to you now is, what about the location of these complexes in terms of synaptic membranes versus non-synaptic membranes, or the extrasynaptic membranes? And what we well, I think I want to go to, I picked no, that is, I wanted to, I must have, yes, this is correct. So what we believe here is that here you have the synaptic heteroceptor complexes coming up over here. And let's say a GABA neuron over here, and the glutamate synapse coming up over in this region. But then we have a lot of dopamine terminals around these neurons as well. And here they are extrasynaptic. located, and you have them all here, integrative activity, and you have an integrative activity here, and so that's volume transmission there, synaptic transmission there. And of course, I wondered, well, how can these things become integrated then? And I felt that perhaps the best is that probably the integration from the synapses and from the extrasynaptic integrative activity will take place in the cytoplasm in terms of controlling the transcription activity in particular, and controlling, modulating phosphorylation, for instance, taken inside here, and then it goes into the nuclei, whatever happens with the transcription. That is just I think it is an important aspect, and I feel this may be a correct interpretation. Then I could not resist to show you this one, because we are very interested in understanding the role of the homo and heteroreceptor complexes in terms of learning and memory. And we believe that learning and memory is based on reorganization of the homo and heteroreceptor complexes in the postsynaptic membrane. And then we also should consider that you ask, well, what about the presynaptic heteroreceptor complexes? Here you have the postsynaptic ones, but you have also integrative activity going on presynaptically, and we believe they are there in order to enhance the pattern of transmitter release to be learned by the postsynaptic receptor complexes. So I think in this way you can have an optimal transmitter release formed, and they can modulate them in a perfect good way the postsynaptic receptor complexes. Also, the question is perhaps so you get a short terminal memory. And perhaps that is not so difficult to think about this way. Things change. The problem is to move it into long-term memory, because how do you get the stability for them to stay around to develop a long-term memory? And I think that the important thing here is that, this is our hypothesis, and the hypothesis we have is that these heteroreceptor complexes, they can be internalized. Some of them can be internalized, and they can then act either as activated proteins or as transcription factors. So they can then target the nuclei, and they can then, the activator proteins can activate them, the DNA, the gene DNA, the enhancer, that can then bend down towards the gene promoter, and then you can have an and then comes the RNA reading of the RNA, and we can have the formation of novel adapter proteins. And these adapter proteins can then bind together. These adapted proteins that can be formed can be cell adhesion proteins, like neuroligands, or they can be master scaffolding proteins like PSD95. So they can bind together and stabilize these reorganized heterocomplexes, and make them stay around through these novel adapted proteins coming up, and you have the potential to develop a long-term memory and a potential molecular engram coming up. And it seems to me that this aspect is for us interesting. I reported this before, but of course I don't know if it certainly has not been criticized so far, but it may be, of course, wrong, but I don't believe so. It makes a lot of sense, and of course it is a good way to you can also here understand lifelong memories through these memories. Why do we can live all our life with long-term? How can we have this lifelong memories? How can that be produced? Well, I think this is not sufficient to explain this. I think it can explain long-term memories, but the stability perhaps is not good enough for these type of proteins to stabilize them, the adaptive proteins. But we believe, or we hypothesize, that lifelong memories may be dependent upon an ability of something very strong, emotions and rewards, which are very strong. And I'm not going to go into details, but you know yourselves that with these strong emotions, they don't go away, and whatever happens, they stay around and around, and till you die, many of them. So I think the reason may be for this is that you get a massive change in the formation of sort of and I'm not going to detail, but the change will be that repression activity in the nuclei is stopped. So it can be stopped via this emotional activation, like the dopamine serotonin neuron can develop a very strong information that can change the DNA in a way that will not allow repression to operate. The repression signal will not be there, it will not be formed. Whatever happens is over the activated proteins, and you get, they will always come through because the repression cannot block them. So the activated proteins, they can see to it that you get your continued expression of the adaptive proteins, the novel adaptive proteins can be kept being formed because repression is gone. It may be true, it may be wrong, but perhaps there is something in it. And so also the bottom line here to me is also that once you start to play around in neurodegeneration like Parkinson's disease, with these receptor complexes being changed and so on, of course, memory will go away. And learning will be very difficult. So at least it's something to think about for the future, I hope. And now I have been spoken too long already. So I will be very quick now. I will try to find something that makes that could be. Well, we don't have to. I think then just the sentence that we went from a very simple story with the one published T receptors. Now we have 15, 16 types of serotonin receptors, and we now know also from our own work, early work, that some should be blocked and some should be activated to reach antidepressant activity. And then I think we entered the various types of the serotonin heteroreceptor complexes came around. And we found quite a lot of them over here. The FABC1, FABC2, the FGF receptor 1, FABC1 complex, and a very interesting complex over here with the galanin receptor 1, receptor 2, and the FABC1A. But I think, and also we have well, I will. So that is, and now I think we switch over to the, well, this is just to say that what we did here on FABC heteroreceptor complexes backed up our hypothesis on the pro-triplet hypothesis, because we found pro-triplet here in these various types of complexes in here you can, with LLG and so on, and to LLG here. So various types of these triplet puzzle theory sort of was supported. But I'm not going into any details. There is no problem. We have time for that. And also remember in the future the dopamine receptor, FABC receptor integrative interactions, which are highly interesting also in relation to schizophrenia there. But this is just the beauty of the past, where we could see the terminals the way without just having diffusion. And I just wanted to show you this one from my thesis. And then I think that we have to go on. And this is just beautiful pictures. The one from the ventral tegmental area, the A10, the dopamine hypothesis. And I think that, and also don't forget the islands, the striatal islands coming up. And what I now want to do is to, the D4 receptors should not be forgotten. Think about them. They are highly relevant, and they have an impact on the striatal and the nigrostriatal dopamine neurons and on the mesolimbic dopamine neurons, with the D4 receptors especially modulating morphine action, so there are dramatic changes coming up there. So I think you should remember this one, and I will just, and this is what we felt, that volume transmission would be a way to to help treat Parkinson's disease, because you know, we felt with L-DOPA treatment, we always felt there wouldn't be enough dopamine that could be formed in those days. And this was in 1979, and I felt that, and perhaps I was wrong, but nevertheless, we did a lot of work on dopamine receptor agonists. We felt that because when the dopamine terminals go away, we believe that this. Sorry about that. Yes, thank you. Thanks a lot. Here you can see that here you have the dopamine terminal, and then in degeneration, of course, there's no terminals here, so there has to be a diffusion over from the dopamine terminals to the adjacent receptors, and therefore, by giving instead the dopamine agonists, it should be a way to improve L-DOPA treatment. And we were wrong, because they are not better than L-DOPA, but they are still highly interesting, and we discovered quite a lot of different types of apomorphine, for instance, etc. But then the one that really made me happy was bromocriptine, because it inhibited the secretion of prolactin, and that was dopaminergic regulated. So we started on bromocriptine as an important Parkinson compound for treatment of Parkinson's disease. And this was in 1979. And I think I would just say that bromocriptine was our best success. It was used at least for 40 years in the clinic finally, and we were even allowed to be part of a clinical study showing the anti-Parkinsonian activity of bromocriptine with anti-tremor actions, but the negative effect was, of course, that could enhance involuntary movements. But we are very grateful to Abraham. Sorry about that. I've forgotten his name there. And then Manny Goldstein for that I could be part of this manuscript. It was one of the first papers showing the anti-Parkinsonian efficacy of bromocriptine in Parkinson's disease. Yeah. And then it was also interesting because this drug, the bromocriptine, did not activate adenine cyclase, but it had a high affinity for the spiperon receptors, D2 receptors. So it seemed to be a novel type of dopamine receptor not linked to the classical activation. That became known as the D2 receptor. And now I think I will go on, and because this approach was the beginning of the heteroceptor complexes in Parkinson's disease, when we found out in a simple model that caffeine and theophyllamine adenosine, that were not even known to be A2A antagonists at the time, but they are. And we found a very strong enhancement of dopamine receptor activation. And that led to a large number of studies, and we became, and like other also group came up with data that A2A receptor antagonists should have a substantial antidepressant potential. And we also found this affinity regulation, well, it also reduced the affinity. The A2A agonist also strongly increased the affinity of the of the D2 receptors, as you can see up here, but also in the human stratum. So that was nice. We had the human correlate as well. And we believed and still believe that this has a high potential. And there are many clinical studies used, but it is all a little disappointing a little bit, the A2A antagonists. They don't give, I thought they would give better antidepressant effects, but they are still around there and it's not so impressive. And I have been thinking about that, why they are not as good as we thought they would be based on the work on the rats models. And we did a lot of work on this. And how come this? How can we try to understand that? Well, we found together with Lars Olsson's group, and we studied locomotion in the Middle Park mice during 8 weeks of daily treatment with saline and the A2A antagonist in combination with L-DOPA and L-DOPA alone. And this we cannot go through in detail, but what was very interesting was, well, when you looked immediately and treated, there were no real differences with A2A and D2. That was not so exciting. But if you look at the 24 hours later on spontaneous activity here, and then this part, this is spontaneous activity, you see that the A2A antagonist has a really nice effect here, and then after 4 weeks, 8 weeks, and 8 weeks over there. And then interesting, it seems to be better than L-DOPA, certainly, in this part. And so the interpretation I felt, this has, of course, to be tested, but this is just an idea that this enhanced effects was when early on treatment, just after 2 weeks of onset of the degeneration. And then it stayed around. So perhaps it is that some of these drugs, like the A2A antagonists, that there is something that goes wrong with the A2A D2 complexes as the disease develops. Perhaps that there develops some sort of binding of another receptor coming in that makes it difficult for the A2A antagonist to act in a proper way and really remove the A2A agonist from the complex, and also its inhibitory regulation. And then if you can keep that going, perhaps you can see results like that. So, or it may be simply that something becomes very strongly binding to the D2 receptors, the A2A. So it sits around there, and it is so strongly bound to the D2 that the antagonist cannot do the job. It just doesn't work so well anymore. It works a little bit, but not as strongly as we would have expected from all the animal experiments performed now over many years. Well, this is what I can tell you now, because now I think time is running out, and you have been here already too long. And we should not forget in the future the oxytocin story. Binding, we found that it had strong binding to the D2 receptors and they enhanced the signal of each other in the dorsal and in the ventral striatum. It was a very strong enhancement, and then I just wanted to state that, and then it also has, so I think we have to consider oxytocin receptors also have a role in Parkinson's disease, at least in terms of social behavior. With those data, they are rather old, but from 2013. I'm not going to this into details, but also 5-HT1A and 5-HT2 receptor can form heteroreceptor complexes with oxytocin, and this was demonstrated by the work was Lekens did the discovery of the complexes. We did just the work on the proximity ligation assay. So I think she did a good job there, Lekens, Harriet. And it's interesting for you to see that you have to bring oxytocin into the picture in many aspects. And when you hear there is inhibition coming up from the 5HT2A and 5HT2C action, and so this is and then well, I'm very good at yes. Well, first of things now today in Parkinson's disease, we are very interested that many of us, you perhaps and others, that we with proximity ligation assay we found the formation of alpha-synuclein A2A heteroreceptor complexes in the dorsal striatum, which made us was quite exciting for us. The green is the labeling of the indicate labeling of neurons, and you see some puncta here coming up. And here you see there, and well, there you don't see them so well. I can see here, but they are there. And what I wanted them to say, why is this so interesting? Well, alpha-synuclein, everybody knows, has a real strong Parkinsonian action in many patients. And what we feel is that if there are many forms of alpha-synuclein, of course, from monomers to dimers to oligopeptides to triplets, high triplets coming up. And in our mind, we have to start to consider that the alpha-synuclein, whatever form, can also be linked to the membrane up here and interact with A2A receptors, and perhaps they can interact with other A2A heteroreceptor complexes and with D2 receptors as well. And so I think looking into the future, I think it will be very exciting to start to look into the action of alpha-synuclein on these heteroreceptor complexes, for instance, in the very important terminal state of parallel GABA neurons mediating motor inhibition, and to see what happens there on these neurons with alpha-synuclein, because the idea is that alpha-synuclein through such interactions may severely interfere with the learning and memory, and therefore the contribution or the interaction between them could be of high relevance for understanding the and develop novel treatments for Parkinson's disease. And then also we do believe perhaps since these neurons they are not so vulnerable to A2A that I just discussed, but what can happen in the site of parallel GABA neurons, they can release alpha-synuclein and together with the A2A, because they form heteroreceptor complexes, and they can then be, they can be taken up into the surrounding dopamine nerve terminals, and which are very sensitive to A2A receptors and also to the alpha-synuclein, and with an onset of degeneration coming up. So this is another aspect. And what I wanted to, in the final slide here, into the future, I think this would be an exciting possibility for the future, this possibility that the A2A receptors and the alpha-synuclein come together, or other type of receptors, who knows about the specificity, and that and also, of course, A2A antagonists can be good to enhance the effects of blocking out because there is a paper published of Ferulia that he reported that the A2A receptor agonist enhances toxicity and aggregation of alpha-synuclein. So this was already written. So we are just moving into the heteroreceptor complexes. I mean, it's not so novel, but the complexes may be interesting to see the vulnerability coming up or not. So this could be an interesting aspect for the future. And I think that what we then could move down here, perhaps, and not go through too many details, is that I realized when discussing this, that you have alpha-synuclein heteroreceptors complexes in Alzheimer's disease may exist. And of course, you all know in Alzheimer's there is a tremendous effect on learning and memory. And this is of course would be highly interesting to determine the beta amyloid, if it can form complexes with also with A2A receptor complexes or other complexes that could strongly contribute to the marked deficit in learning and memory in Alzheimer's disease as well. So the future looks interesting. We may be at the beginning of something interesting, and it seems to me that the heteroreceptor complexes will stay around for some time because it offers novel integrated possibilities and understanding of brain disease. And the last thing I just would like to, well, I'm sorry, but I wanted to just to read to you one line, and that is that in relation to the discussion ongoing now today, and we do believe that the introduction of the receptor-receptor interactions was the origin of the concept of beta modulation provided by Katz and Edwards in 1999, which stood for the fine-tuning or modulation of neuronal transmission. However, I believe, or we believe, that another term should also be considered, namely protein, or I would say actually second-order protein modulation. Second-order protein modulation, because that is, in essence, of the heteroceptor complex world. And we do believe that allosteric receptor-receptor interactions will, and their involvement in brain disease and its treatment are of high interest. And their pathological relevance has been indicated, especially for major depressive disorders, Parkinson's disease, and cocaine use disorder. Thanks a lot for listening. I'm sorry to be so long. Thank you. Thank you.
Bueno, pues vamos a tener ahora una breve sesión de preguntas y respuestas. Les pido disculpas de la práctica que alargó, pero fue no por causa del doctor Huxley, sino como ustedes vieron, por causa de ese incidente que tuvimos. Pero bueno, con la plática y ahora me gustaría oír la interacción con la audiencia. ¿Quién tiene alguna pregunta? Paco.
Is it known if the heterodimer interactions are modulated by the lipid composition of the membrane?
Could you come closer so I can hear, because I don't hear so well?
Is it known if the heteroreceptor function is modulated by the lipid composition of the membrane?
Well, that's a very good question. I think this is unknown. It makes sense to me that they could be modulated through the lipid environment, but nobody knows. There has been no reports on that, but it's a good question, so thanks a lot for that.
¿Una otra pregunta? Bueno, yo tengo alguna pregunta, y esto tiene que ver un poco con la pregunta que quiero hacer. I would translate it.
Okay.
I have a question, and the question that I have to do is that receptor-receptor interaction has a lot to do with, say, topology. Because, of course, if one ligand goes into one receptor, then this receptor, when it's activated, should meet another receptor, which will be close enough to make the whole thing working. So is there something known about, say, for instance, topology among receptors within the brains?
In terms of the topology, you mean distribution pattern? Or? Yeah. Well, of course, the distribution pattern is fundamental. This, of course, is true, and that's why you have to be so careful with cellular models, which I like and love. But in the cellular models, you just, put them in there and you get breadth to fret, and there is an interaction or that. But nobody knows if it happened in the brain, because they may not be collocated as a starter, and then it was not official interaction. But on the other hand, I do believe that in most cases there is a perfect agreement. But I think I remember some places, some indications that they were not so clear. So it is, that's why in this type of research, as you indicate here with your question, excellent question, that we have to do both cellular models together with animal experiments, and at the end finally reach the human brain, which is extremely difficult because we have tried that, but it's not easy. And that is the optimal way to go ahead in a way. And yeah, that's it. I think. And of course, you can always match and that's important with immunohistochemistry. You can match the PUG that had come out in red and see the the orientation with labeling the dendrites with markers and also the glia and the entire neuronal network with to label them green. So it's a lot of efforts, but definitely if I'm interested in integration and understanding to improve treatments of disease, mental disease and neurological disease, it has a potential, this technique. It has a potential.
Okay. Still, receptors are being thought, I mean, GPCR receptors are being thought.
Are being done?
Thought. Yeah. In the books, yeah. In normal books, cell biology books, pharmacology books, as monomers and signaling cascades for signaling cascades.
Yes.
Signaling cascades for each receptors are being described as canonical. Yes. Alpha 2, alpha 1, beta 2. And this is how they are taught in the school, in the book, textbook.
Yeah.
How, I mean, much of this work was done in cell lines.
Yes, well.
Express it, you know, when you express one receptor. However, in the native tissue, what makes you suspect that you are having an oligomer? You do an electrophysiology experiment or biochemical experiment, binding experiment. What makes you to suspect that you don't have a canonical signaling, that you have an oligomer?
Well, I mean, you can have both, of course. I mean, it's complicated, and you then if you believe that if you see it in the cell line and you see and in the brain, you show it with the proximity ligation assay is very good. It's a very sensitive, excellent method. So, and you always have control animals in everything, like you have controls in the cell lines. I mean, without controls, you're out of business, of course. You don't know what you're doing. But then with all these controls and the proximity ligation assay, but then it is also true that we may not see everything with the proximity ligation assay, as a matter of fact. It may not be sensitive enough. So far, it has always been correct because you have so many controls. It is true. But you are right in a way that there can happen things that means that some you cannot discover, for instance, because they are this dimer at another place. It's interacting with another receptor, so the dimer becomes a trimer, and you cannot pick them up anymore because it's all changed. Reorganize is change, and then even if you have seen them in the brain, but then they can become alterated. So then you don't know, and they're just around, they're close to each other, but they don't interact. You don't have a puncta coming up, and you can demonstrate that with novel techniques also, as a matter of fact. So I think, but I don't know if I've answered your question.
For example, if I make an experiment, and I know that the receptor is such and such, and I expect a signaling cascade, and I do the test, and there is not that signaling cascade.
Yeah, okay. But
is that a signal that maybe we will have just the monomer of the receptor, I think? is that a signal that it can be forming an heteromer?
Well, I don't. The basic mechanism from a monomer to a heterodimer is not. We have not seen that in our work that suddenly that property will disappear. But it varies, of course. Sometimes it can you can simply you can there could of course be but if there is a change, you pick it up with PLA that's relevant. But you mean that and of course the signaling can become altered, of course. This is what we have found. So when this signaling they had can change, if you go from a monomer to a heterodimer, of course you have an allosteric receptor interaction that may kill that GS coupling or GQ coupling, and you get another type of coupling instead coming up in that receptor that previously, let's say, were operating via GS. Together, when you go into the heteromer, it becomes involved with GI or GS, or the GI or some other G protein coupled activity. So or beta arrestin, yeah. So the story is it's very plastic brain. There is no just silent sitting around and not changes are not developing. So I think the bottom line is tremendous plasticity brought about through the allosteric receptor interactions, and the signaling is affected. The selection of compounds is changed. Suddenly they can go from localizing, if you get the heterodimer, this is with the galanine 1 and galanine 2. You know what they like then? They don't like the galanine itself anymore. They prefer the galanine fragment 115, just as an example. And then, of course, the affinity changes, and also the density of the complexes can change very much with agonist antagonist treatment of different types. So the bottom line is plasticity through the allosteric receptor interactions, involving also and it involves. And we've been talking now about the GPCRs all the time, but we have GPCR with RTKs, which are very powerful, modulating the plasticity in a great way. And of course, they have different type of signaling because you develop allosteric interactions that blocks part of the story and explains the data that this is a disease, can help understand this disease development in for instance, depression, etc. So the key is use proximity ligation assay in a good way with controls is a good starter together with also analyzing the modulation of affinities and Bmax values, and it is one reason is that these complexes offer a new way for understanding what happens in the functions. How can this go on? How can this happen? This cannot be wrong. And then you go down to it and look at the complexes and see how they can be really markedly altered, many of them in new directions, and which can be described in various ways. And it is simply the bottom line is that this, I call it receptor modulation, is a very important term in my mind, because that's what's going on. And it is plasticity, and it is, of course, especially in higher order complexes. And it has helped me to understand a lot about functional aspects in behavior experiments of various types. Well, we do it. You have a change there, so it can explain these events. So, but you have to be very critical and absolutely use proper controls and then the controls. So you, otherwise you have to get it back anyhow to revise it, you know. So that is the situation. And this and I'm happy this concept came around, because I understand the brain much better now. This is the way it is. So yeah, I don't know if any more questions.
Is there any other question?
No.
Please, ladies first.
Thanks. Well, I am very interested in plasticity. Is why I am studying neuroscience actually. And well, I investigated about plasticity, but I found that basically the theory is that a patient has abilities, but he or she not use it until he needed by a lesion. So I was thinking if the plasticity for treat the Parkinson disease and Alzheimer's disease is take disability inhibited and disabilities that are inhibited because the person in a normal state don't need it and elevate the activity of disabilities, or is more than separate the complex formed by E2ER with alpha-synuclein.
Yeah, I did. Well, so you feel, you wonder if could you repeat the question again?
Yeah.
I think she's asking if during plasticity, if there's something you are not using that's inhibited and it can express under different situations.
Yeah, I read that a normal person has abilities that used in a normal state, but when he or she started to present a Parkinson's disease or Alzheimer's disease, the plasticity can help us to take these abilities that was useful and measure the activity, start to use these abilities that in a normal state the person didn't use it. But my question is if that's I don't know how to say it. If the plasticity can separate the complexes created by A2ER and alpha-synuclein.
Well, yes. Well, of course, plasticity can have a major impact on the changes, of course, that we observe in these with these potential alpha-synuclein GPCR interactions. I think you are perfectly right that the RTKs, receptor tyrosine kinases, here have probably a very important role in participation of the modulation of these events that goes on with A2A receptor, with the for instance with the alpha-synuclein and so on. And of course, the question we have now is, to which extent should we focus on the GPCRs? Perhaps we should focus more on the or as much on the RTKs, and perhaps they have a really important role in this case. And I do believe that there are several examples from Greengard. Greengard actually he came first with giving evidence for the interactions between the A2A and the receptor and the FGF receptor 1. And that was a dramatic interactions they demonstrated in normal rats. I mean, they have no models. They're just using various types of cellular models in particular. But there were dramatic changes coming up. And perhaps when you move into neurodegeneration, like we said, when you move into Parkinson's disease, and when you move into Alzheimer's disease, I think you're perfectly right. You shouldn't do it without at the same time considering the role of the various or the most interesting types of RTKs participating and how they are altered by the alpha-synucleins as well as the A2A receptor. So this means that you are perfectly right there. We should also include the like FGF receptor 1 or other types, whatever seems to be most relevant in the area you're analyzing and studying. So I would certainly do that. And then of course we have a very exciting results over quite a few years now on FGF receptor 1 and the 5-HT1A, which gives just beautiful data in models of depression. But there was no time to speak about this now. And this is another example, and I do believe that when you move into alpha-synuclein, into the Alzheimer's disease, I think you should have a good balance with the tyrosine kinases receptors. They are very important, and we know that GPCRs modulate also the RTKs through receptor-receptor interactions and heteroreceptor complexes. And neurodegeneration, perhaps they should be together with A2A, be in focus. And do that, let's say, in the striatum, in the dorsal and ventral striatum as a beginning, and then, if possible, then later on could also be going, then move over to nigrostriatal as well later on. I think there's a good future there of understanding, more understanding of Parkinson's disease and its potential treatment. And I think that there is no doubt in my mind that when you speak about the neurodegenerative disease, I mean, you have to remember these heteroreceptor complexes with focus then on the RTKs and A2A receptor in particular, and that is very important. That and now I realize I didn't even discuss ionotropic receptors like NMDA and everything. So you will say, well, this is a crazy guy. Well, you cannot do everything at one time. You have to do what you would give to your own brain the priority to. But you are absolutely right. Thanks a lot. Plasticity and RTKs, that would be a very important addition to the GPCR stories in complexes. Thanks a lot.
Okay. Well, thanks for your time, for your presentation, and it's an honor be talking to you, and that you will answer my questions. And sorry for my English, I have a lot that I didn't practice.
Okay. Thanks a lot.
Is there any other question? Well, I think that we deserve a good lunch. So it's time to go to it and enjoy the meal.
Thanks a lot for the questions. Thanks a lot. So I had forgotten the article.